much for the great introduction, although I wrote it, so. That's me writing about myself. I'm going to be giving a talk about the collaborative river biomonitoring approach. And this part of the title of my talk today will come midway of my talk. So I'm going to present you what we are doing in the Mara River, what we found, and what inspired us to really focus and move forward with this approach where we're going to be using community to try to monitor the river health in this river and having ideas to even upscale this from the local scale to even like a national focused kind of a monitoring thing. So to start off, is a lot of you in this room will agree with me that globally there's this trend where we're losing a lot of our biodiversity, and there's this kind of a trending word nowadays that we need to bend the curve, right? So the bending the curve is literally just moving away from the biodiversity decline and trying to control that with whatever approaches and what scientists are learning out there. And that's what inspires my dissertation or my chapters. I'm more into I'm in biology, but focusing more on conservation aspect, trying to protect species and things like that. And I'm going to be taking you through the journey of what we are working in the Mara River, what species we are protecting, and what we think is ideal working with the communities there. So we've seen that the trend of this biodiversity declining, but also if we compare these two between the terrestrial and aquatic or freshwater species, we've seen that the trend with the freshwater is even further declining as compared to their counterparts, just because of these prevailing other threats, which are mostly in freshwater, things like all those emerging threats nowadays, climate change, talk about invasive species, talk about land use change and what it does to these aquatic systems, and all this nested away of, what really affects the freshwater ecosystem. So the thing is that the freshwater are more threatened compared to their counterparts in terrestrial system. And if we went further and looked on this global map and see how things are happening, is that has been a country somewhere here along the equator, the tropics, where Kenya is at, and our Mara River is there, our aquatic systems along are there, that we are foreseeing a trend where in future we might even be more threatened, this ecosystem, the freshwater ecosystem, due to things like climate change, temperature increases, and things like that. And so you might be wondering, why the Mara River? Why not any other river in Kenya or in the rest of the world? Is this because this river is one of the important transboundary river between Kenya and Tanzania, and this year is where we have the famous migration of the wilderness. Those animals which looks like our bisons, those are the wilderness there, and we have this annual trend where they are migrating from Tanzanian side all the way to the Kenyan side. And the river they have to cross when they are doing this migration is this important river called the Mara River. And as they do this migration, what is happening is that at some point we have tons of them drowning in the aquatic system, in the Mara River system itself, and we have them decaying. There are thousands of wildebeest carcasses just on the riverbed, just decomposing. Vultures are coming, feeding on those wildebeest. All those nutrients are leaching to this aquatic system. What does that annual event then do to this important river system? Apart from that, this is the same scenario during migration. Everybody is like going there in this savanna and seeing what is really happening there. So it's like one of the most touristic area because it's the ecosystem between Masai Mara in Kenya and the Serengeti ecosystem in Tanzania. It's a river with mega herbivores, the hippos, we have crocodiles, and we have really also like big fish species in this aquatic system. It's important river for water for people there. That's where they directly get their drinking water from, and especially the local community, the Maasais are literally just fetching this water from this river, and that's what they are using in their household, even without undergoing any form of treatment. So that's why the Mara River is there, and you're like, why not another river? And in this system, what we are mostly interested in is that we have the interplay of these three different stressors. That land use is really a thing in this catchment. It's documented. It's been happening. And what then does it do to this river, this aquatic system of the Mara River? Things like point sources of pollution. I showed you an image where it's one of the most touristic places in the world during this month of July and September when these wildebeest are migrating. What does that mean to this river in terms of the launches being built? Where do they dispose their waste to? Where do these wildlife subsidies from these dying carcasses, the importance of these hippos in the system, what does that mean to this river, this important river there, and these communities of macroinvertebrates? So I'm going to be showing you in a few slides what these organisms are and why they're important and why the communities in this ecosystem are using them to monitor the river health. So I take you through a journey where this river is at. This is the map of our study area between Kenya and Tanzania. This red line is the transboundary border between Kenyan side and Tanzanian side. And for now, our sampling has been more focused on this Kenyan side where we longitudinally sampling array of sites along that river. And this is how the land use looks like for the ESA. This is data from ESA 2021. We use their land cover classification to come up with this map. What is seen here is that the upstream sites, mostly these M1, M2, and M3, are within this our only protected area, which is the major catchment of this area called the Mau Forest. And Mau Forest 15 years ago, the communities were somehow allowed to live in this forest. Then the government realized the mess they were doing, and everybody was pushed away from the Mau forest. That has been like less than 10 years ago. And the government has already compensated them, but people are there and are not, happy that they were moved away from the forest because the forest was more productive. They were doing a lot of farming, but what does that mean? We're cutting more trees down so that they can farm where they're getting a lot of food. It brings more here than anywhere else, as I can be showing you in the next map with the rainfall trend of this region. But in summary, is that this area has a little bit of forest remaining. We have lots of grassland. If you follow the map with the cropland, you see how also closer to the main forest where now people are at, there's a huge agriculture going on there. What does that mean in terms of pesticides, fertilizers into this catchment area? Built up areas, they are in reds, probably not seen now, but these are like the towns along this river and things like that. So, and this is the map of the rainfall for average for the past almost 20 years in this region. And what we are seeing is that the area up here, which is our upper catchment area, is receiving more rainfall over the 20 years as compared to these other sites along this catchment. And so we use an array of sampling techniques in this region to be able to collect or capture what we are calling the macroinvertebrates, which are basically these smaller organisms. Sometimes you could see them with your naked eye. Sometimes you need aid of microscope to be able to visualize and see their morphology. But in the better part, you only could be able to even see them with your naked eye. And those are the organisms which are living in water during their larval stages. So a good example is that an organism like a dragonfly, you all relate what a dragonfly is probably is and how it looks like. So that's also categorized as a macroinvertebrate because it spends part of its life during the larval stages in water, then it grows wings, and then as an adult, it can live out of the water. And these are some of the examples of these organisms you're referring to as the macroinvertebrates, or if you like, the benthic macroinvertebrates. These three at the top are what people call them an acronym. There's an acronym called EPT, which is basically an acronym for mayfly, stonefly, and caddisfly. That's a mayfly, that's a stonefly, this is a caddisfly. And these three, from the literature, from everywhere else in the world, people use those three to even do biomonitoring in rivers, because if you find them in some sites in a water system, they can tell you a story. Like if you found only those, it infers that, oh, you are dealing with really good water quality here. Everything looks good and there is no cause for alarm. But if you found these other category of these minches, these are chironomids, and these are the black flies, then that sends you a signal that, oh, the system is somehow changing from this good indication, and now it only harbors these organisms which are not really good indicators for water health, and all that's what that means then. So I'm just going to take you through what inspired us to come up with this approach of trying to use communities and monitor the river systems in Kenya through what we found already, because a lot of research inspires the next steps. So I'm going to show you what the results are already, what we found, what is inspiring us to use the community for monitoring. So over time, this is one of my bigger results, main finding. You've seen this for the first time before it gets published, but we are in the process of working on that. And what you are seeing here is a PCO ordination, and basically we compared data from 15 years ago and now. 15 years ago was when my advisor Amanda Spalavsky was still working in the Mara River system. We had collected all these different samples from all the sites I'm interested on. So we have that data, and I came in my PhD, studied in 2021 until now, and we have the same data. So this is their data, and this is my data. How do you interpret that? So historically, is that the sites were almost looking similar. All what you found in terms of species were not so different from each other in all these drill sites. Currently, what I'm finding is that these sites have further moved from each other. Things have become more different, and we could even quantify this statistically that, oh, there's a change between the historical sampling and the current sampling. Things have changed over time. And this change was even significant when we tested it and all that. I don't want to bring a lot of statistics here, but what is inspiring us to do this is over a period of 15 years, based on what stressors are happening in this catchment, we've been able to demonstrate that species have changed. There's been a complete turnover in these different sites. Is it a good turnover or a bad one? So that's gonna be the next kind of finding we are. And also the diversity aspects, also the species in terms of diversity, if you compared the black boxes and the orange or red boxes for colorblind people like us, you'll also see there's been a significant change between this diversity from historical to now. And that was actually really significant as well in the same sites. Further, we wanted to see what was driving these changes in the current composition of these communities, and we found a very strong correlation between these 5 environmental variables. We compared 14 of them, but between the 14, only those 5 were able to stand and tell a story that things are changing currently because of differences in temperature, differences in nutrients, in water, due to differences in dissolved oxygen within your sites, differences in elevation, as altitude is a good proxy, or it's a good kind of a thing which explains the distribution of species as well, and also the flow of these rivers as well. So those were the 5 main important factors in this river. We went further and compared our EPT, our mayflies, stoneflies, and our caddis flies, because these are known to be good indicators of water quality. How then are they doing within our catchment? If you sampled this river, do you find them everywhere? Where are they at? Would you use them for biomonitoring aspect? And it's so clear that as you move, so our upstream is somewhere here, because the higher you go, the elevation closer to your water catchment area. It's inverse true, actually, what we are finding, that these important sensitive taxas are actually declining as you move further to where the water comes from. It could have been the other way around, and I'm still struggling to interpret these results. A good graph for me will have been if this graph was kind of increasing, but now it's telling you as you move further from where we are calling pristine, is where the abundance of these organisms is increasing. Does it mean that they want to be there? Have they adapted? in this kind of water system which is not good? So those are kind of questions we are trying to answer, but you could ask me follow-up question on that. And so that was all about what we have done in this system and what we have seen. And based on our results, then we were like, oh, okay, things are changing. And could we really tell a story using these invertebrates then? There's this technique by the name South African scoring system, which is basically a document which has been used over time in African countries, the ones who are using these organisms to monitor river health. And what this document does, it has a protocol where every organism, every single of these invertebrates has a sensitivity score. So if you found it in a certain river system, the sensitivity scores are always like, constant numbers. So the EPTs, which are good indicators of water quality, the indices is between 1 and 15. The higher the number assigned to every individual, the better its sensitivity to tell you a story on water quality. The lower the number, the more tolerant it is to pollution. So this technique, I've always wanted to learn how it's done. And in 2021, when I started my PhD, I was lucky to go to South Africa and I got certified in this technique. So like, it's something I've been trained on, it's something I could use. And actually, I'm now part of those people who are now certified. And this means even if somebody wanted to contract you in South Africa to conduct this kind of business, you are eligible, I could do it. So this protocol, these are the numbers. QV means the quality value for each organism. So you have a checklist of all the possible organisms you can find in a river, and each organism has its sensitivity score. And using this protocol, you are able to calculate what we call a SAS score, which is this. SAS score is basically all the present individuals you found, you add those numbers, and that's what you get as a SAS score. The number of taxa is basically how many different species you found in every site. And if you use those two indices, then you could calculate something called average score per taxa, which is basically your SAS score divided by that number, and it gives you a value, all right? And using that value, you are able to calculate or tell a story about what is happening in your catchment. So we used our data from the Mara River, which was more historical from 15 years ago, and the data which I had. And one take-home message is that when you use those numbers, the numbers you could interpret them using a graph, where the graph will have the final numbers you calculated, the average score per taxa, and your SAS score. And using that graph, you could plot. And when you plot the numbers is when you get these different categories, which are already known. And when your data lies probably at A, this is what it means. When it's at B, all that is what it means. So fast forward is that if your data lies in A class, that's what it means. Like it's more like a natural state, is unmodified kind of a system and things like that. So we did this for this river, and we found like historical sites 15 years ago when we did this. The macroinvertebrates are saying that all these sites, actually good sites, they are storing an A, which is almost like a natural state, more like pristine. Fast forward, in the current year, when I've done this between 2021 to the present year, is that only one site retained its integrity. The rest have somehow lost the integrity from an A and they have moved to a B now, all right? So it's something you actually use, and this is basically what I started with. This was more similar, and things were more like scoring an A. Things have moved apart, and that's when you even like could calculate and see, oh, water quality over 15 years has changed, and now our sites are not natural anymore. There's more pollution in the catchment and things like that. When we plotted this is our historical data, this is the current data, and you could see, oh yeah, things are actually shifting from what is a good natural site to even becoming more and more bad over time, right? And with that, we were like, okay, so far, what we've seen is that with everything pointing out, there's no way we're gonna save the Mara River or do everything or even save the rivers in the world without collaboratively working with the people who benefit from this river system, right? Who is encroaching this water catchment? The community. Who is the beneficiary of going to this river and fetching the river? Who pollutes this river? there are these launches, there are people in the community, then how then could you work with these people and prevent the levels of what you are seeing currently in your catchment? And this is now what took me to Kenya this year. I was in Kenya between January and August doing my TCD practicum. And what I wanted to do was to involve the communities and train and build capacity in some of these institutions, especially at university levels, because where we do research, there are also universities around there. People are probably not aware of what is happening. People are not aware of the harm the Mara River is faced or any other river around. People are not aware of what these invertebrates are and how could you use them then to monitor a river, you know? So there's a lot of moving parts, and I was like, since I'm trained on this, I want to be the voice out there. I already have the capacity to be trainer of trainers that could tell other people on how to do this. So what we did, because we wanted to work with universities. So what I'm going to be showing here is what we did with universities back then, from January through April. And basically, we wanted to do our session in threes. So what do I mean by three parts? We wanted to have a seminar with these students. If we were given a grade note by the university, we wanted to tell them what the overall picture was. What is this research we found and what are we finding? And we wanted to even them have to feel what these invertebrates are and to embrace what these organisms were. So we were doing a seminar. We were asking them out to do a practical activity down in the river where we go sample these invertebrates with them, bring them back to their labs. We could also provide them with identification tools, and we could all together identify these organisms. And lastly, I was telling them the story of the applicability of this tool, how we could use all these different techniques to even carry this initiative on their own, because a lot of these universities have environmental clubs and they tend to be next to a river somehow. There's a lot of these universities. So the environmental club could lead such an activity even when I'm not there, you know. They could be doing this like semi-annual, bi-weekly, monthly, who knows? And they could even have data on how their river is doing in terms of health. So I went to these three universities. One was Egerton University, where we did a seminar. We went with the students to the river, showed them how the technique works, how do you need to sample your organisms, what kind of protocol do you need to follow. We came back to the lab uh, with the head of the universities who are really excited, and they are always providing these facilities for the students. They will provide transport to our nearest river to go sample. They will make sure the labs are ready. Even we did this a lot during the weekend, so they ensured everything ran smoothly. And I was a lot of success I had with these universities is somehow I've been in one way or the other interacted with them. They are either my alma maters, institutions where I went to school to, or I collaboratively work with some of the professors from those universities. That's why I had success in these three main ones where I did that. So that was that institution. We went to Jomo Kenyatta, which is also another bigger institution in Kenya. We did the same thing, giving a seminar about all this, what we do, going to the river, bringing the samples back to the river. And I also forgot about the other part of the seminar. We're also doing more like a mentorship kind of a program. After now, all these how this all science works, a lot of students are curious, like, how can I get to follow your career path? Where do I start? If I'm doing a bachelor's, where do I then? What kind of master's program should I be looking for? What are the opportunities out there? So we had a session as like that as well, and that's the other last institution, which is Karatina University. We also did the same thing. The students were really happy about all this procedure, the training, a lot of these staffs I was working with in correspondence with the emails. There was some great feedback after the training that everybody was really happy about this training. I've been reached on severally by students who wanted me to guide them on how to apply to these kind of scholarships, which they could follow a path in what I was doing. They saw it's really cool. The institution were really, really, thankful of what I was doing there in Kenya, just getting my time and training the fellow students free of charge and just like building their capacity and all that. And so, as part of my practicum, I was also able to interact with some of the communities there. The Maasai community, here I am. I'm being assimilated to be a Maasai. This is the river where we work in Masai Mara, and these are also the people you need to talk to because they are the closest beneficiaries of this river system. These are some of the stakeholders who really you need them to interplay to protect the Mara River because here this was a meeting with this is like the chief officer of the Mara Conservancy is like the guy whom you want to meet when you have to do anything in terms of conservancy. There are a lot of all these guys are brought all together, the different other stakeholders or actors who are doing different research in the Mara River, but also keen on what is happening on the Mara River because Mara River is like runs their business as well, so they want to be part of the conversation. So we had also some seminars talking about the Mara River and things like that. And next step will be this year I've been awarded this fellowship by the WWF Russell E-Train Fellowship. It's a great one. They want me to continue doing what I did during my practicum back in Kenya. So next year, I will love to upscale what I was doing at the university level with these other different levels of institution in Kenya. I want to work with elementary kids. I want to work with high school schools. I want to work with the community-based organizations, and I also want to do other little bit of other universities who are keen on the same thing in Kenya, and not just on Mara River, but we're gonna have to upscale it to like a national level kind of a thing where everybody is on board. They know the importance of the river system. You need to do this to protect them, but also you could use these important indicators, which are invertebrates, to monitor your river system. Uh, During my stay there, I was also fortunate to attend some of the conferences. This is like story of my life, what I did all that time. There was this great conference I went to the UK, to Cambridge. Student conference on conservation science happens every year. Their deadlines are usually around, I think they're open now. The conference is usually in March of every year. They have a version of the UK is the main one. They have another one in New York. Belagras. What's the name of that country? Am I saying it right? Belagras. I don't know what I'm saying, but there's another version somewhere in Asia. And so what is happening, it brings all the students doing research in different fields and just tell their stories. You could do a poster, you could do oral presentation. I was fortunate to meet this guy. And this is John, not Dane. John Paul Rodriguez is the chair of the IUCN Survival Species Commission and introduced me to that commission, which now I happen to be a committee member of the IUCN specialist group in the Species Survival Commission, both the global one and also at local scale in Kenya. I'm involved with the Kenyan chapter of the Species Commission thing. We also are able to go to HBC in Kigali, Rwanda. Probably many of you know who she is. I can't recall her name again. Leandra. And a few other guys from UF. You also know Audrey and another colleague of mine from biology department. So this was like a UF kind of a thing at HBC, which we are thankful to, of course, TCD always funding us and providing support to attend such meetings. And since I work in Kenya and Tanzania, this was our first time this year to be granted permits to continue with the work we do in Kenya. If you saw my map was missing some sites in Tanzania, we now have the permits. And this is an interesting lower stream of the river because what you're seeing there, these are massive pipes. What are they doing there? Where are they coming from? So those pipes are emanating from artisanal, not artisanal. Actually, this is like industrial scale gold mining in Tanzania. And this is their waste, which is like treated waste, but of course contains remnants of mercury and other kind of trace elements they use when they have to process this gold and all that. Everything gets into the water, and this is a few meters just from the pipes. Then we have the community members who are drawing water for their homestead use. They are washing their clothes. These are actually some cooking pots. They are washing them next to the river. This was like there and here. So all that is happening, and downstream, there's a lot of activities happening. People are using water, fishermen are doing their thing, and you know the fish and all those other organisms have the potential to accumulate these toxicants from being released in water into their body. And that's like another chapter of my dissertation where I'm looking at the effect of ecotoxicology in this river system, both in water, column, sediment, in fish, and also in these organisms called macroinvertebrates. So we had a great time there with my advisor and everybody, collaborators, and it was our first time to actually sample the lower section of the Mara River. So that was awesome and great for this year. And all after I do these tasks and hard work, I have to repay myself sometimes in Kenya. So I had time to even do what I love. I usually do the annual hiking of Mount Kenya itself. The altitude is pretty high. This was 4985 meters above sea level. So this is like our second highest mountain in Africa after Kilimanjaro. I also love to play soccer sometimes, not sometimes, every time. And when I'm not doing my science, you'll find me somehow getting integrated in the community, either hiking or doing something I love to do. And with that, I love to acknowledge my beautiful team there, researchers from the National Museum of Kenya, my advisor with all the support always, Amanda and Chris, and this different funding organization, which somehow they funded my work all through from 2021 until now, both external grants from Explorer, Ruford, them a few, and of course to our amazing TCD Center for African Studies with the dissertational grants and other bodies there and there, and the recent grantee, the WWF Research Train, and always these amazing guys for always giving us permits to undertake our research in Kenya. And now I open this space for questions.